Correct. All right. Anyway, Sullivan's not here. Hill is online. Byrne is not here. Tyrus is here. Lacey is online. Fitzpatrick is online. Thornabar is here. Huffman is not here. Hamlet is not here. Johnson, not here. Bayard is not here. Sigmund is not here. Michael Baumgardner, not here, not yet. Anyway, Pena, not here. We've got Hamrick and O'Brien are both physically here. Ray is online. Davis is here. Yes, Davis is online. Lloyd is online. Elias is not here. Breeden, not here. Cooper, not here. All right, did I miss anybody? It's just now 8, so I'll just do announcements before we get started. So one of your classmates asked a very good question about the lab. More specifically, the lack of the fact that, I haven't shared it yet for this coming lab, this coming week. And that's because I'm still deciding. So definitely the rest of the labs will be online. And they're almost all definitely the next 2. There's only 2 left. They're almost all definitely going to be videos. So this week's, the one that starts on Wednesday, that'll be due next Wednesday, it is going to be a video. I'm just trying to narrow it down to which one I'm going to do it about. So you're going to have a video. It'll be a documentary somewhere between 1 and 2 hours. Definitely less than the length of lab, because lab is 1 hour and 50 minutes long. So it'll be shorter than that. I can promise you that. And that'll be it. So there's no pre-lab, nothing like that. It'll be really simple. It's the end of the semester, so I know a lot of people are really busy and have a lot of things to do and a lot of studying to do. So lab will be easy from here on out. So any questions about lab? All right. Exam grades, keep an eye on the Google Classroom. I'm still going through them grade, person by person. I'm nitpicking every single question, trying to find ways I can add extra points. So I will let you know when the grades are finalized. I'm almost there. I shouldn't say I'm almost there. I think I've gotten through about half of the class. So half of you I've gone through and given you as many points as I can. I just need to do it with the other half, at which point the grades that are on the grade sheet will be official. And also at which point I'll decide whether or not I want to let you guys redo it again. And it looks like right now that'll be the case. So remember the first exam where you took it, and then I said, all right, you can take it again, and then it was an average between the 2? It looks like that'll probably happen again this time. So you took it, that's your original grade, you take it again, and then I'll average the 2. Unless, of course, you take it again and the grade's lower, in which case we'll just keep your higher grade. So there's nothing to lose when the time comes, if it comes. So any questions about labs, exams, anything else? All right, let's get started. Here we go. Back to evolution. I need to get a move on. I'm being a little slow. But we get to this end. The end is my favorite part of the semester. Is even though I did my thesis work in genetics and looking at cells, what I really love is this big picture stuff. I love evolution and ecology, how different organisms interact with each other in the environment. Anyway, this is where we left off. We were talking about the Hardy-Weinberg formula, which you're already familiar with from the lab. And again, I want to remind you, allele frequencies, again, this is assuming you have a gene that has one of 2 alleles, right? Like the chin dimple or the eyes, according to the lab we did, or freckles or hitchhiker something, whatever, right? You only have one of 2 alleles. So allele frequencies is P plus Q equals 1. So the dominant plus the recessive equals 1. So 50% of or 0.5 is Q, then 0.5 must be P. If 50% or 0.7 is P, then 0.3 must be Q, right? Because it all needs to equal to 1. Same thing with the genotype frequencies. And I think what usually gets people thrown off about this thing is it just looks complicated. Unless you're into math, you might look at that and be, man, I hate algebra. Screw that thing. But try not to look at it that way. Just try to look at it as P squared is the number of dominant homozygotes, right? So big A, if we're using A's. The 2PQ, those are just your heterozygotes, right? So your, big A, little a. and your recessive homozygotes, that's Q squared. So that would just be, the number of people in your population that have little a, little a. So again, I know it looks confusing, but remember, what we're just talking about is when it comes to genotypes, when you have a gene that only has three alleles or two alleles, that's it. You're only going to have homozygous dominants, heterozygotes, or homozygous recessive. Those are the only three people you'll have in a population, or things you'll have in a population if you're talking about flowers or whatever. And that all just equals to 1. That's all. That's all that's saying. That being said, there is a more complicated reason. This isn't just for looks. Yes, it truly is algebra. And the next thing I'm going to show you is going to demonstrate that. But your book includes it, so do I, obviously. And I'm going to explain it. I'm going to go through the math with you. But this whole time, I just want you to look at the big picture. Don't focus in on the details, especially if you're not good at math, because you're just going to get lost in the sauce, and I don't want that. This is just going to show you how we can use the Hardy-Weinberg formula in public health in this example. So again, don't worry about this. I'm not going to ask you to do this. This is just an example. All right. So this is a real example too. There's something called PKU. PKU is the inability to break down a certain amino acid, phenylalanine to be exact. This is caused by a recessive allele. And to remind you, and this is important, this part of it, remember, if it's caused by a recessive allele, that means you have to have both recessive alleles, right? You have to be homozygous recessive to have it. Anyway, it's caused by a recessive allele. We know that 1 out of 10,000 babies in the U.S. are born with this disease, PKU. With that being said, we can do some algebra. Now go back in your mind. I'll just write it here. Hopefully you guys can all see it. P squared plus 2PQ plus Q squared equals 1. So I'm gonna abbreviate here. Homozygous dominant. Actually, you know what? Forget that. Let's just use letters. I'm going to use, T just as an example. Homozygous dominant, heterozygous, and homozygous recessive. All right, just as a reference. And again, we're talking about homozygous recessive. So we know that 1 out of every 10,000 babies has this. So 1 out of every 10,000 babies is Q squared. Now, how do you write 1 out of 10,000 in the decimal form? Well, it's just 0.0001. You with me so far? That's what that means. That is the same thing as saying 1 in 10,000. So if you take 1 and divide it by 10,000, that's the number you get. Now, we're going to do some algebra. We want to get the Q, first of all. So if Q squared equals that, we just do the same thing on both sides, in which case we're going to take the square root. Obviously, the square root of Q squared is Q. But again, we did it on one side, so we have to do it on the other. The square root of 0.0001 is 0.01. So Q equals 0.01. And if you remember, P plus Q equals 1, to remind you, right? So Q equals that. Let me show you this. Again, I apologize for all the algebra. If P plus Q equals 1, then we know, let's see, in this case they went with the P. If we subtract a Q from this side, then we get P equals 1 minus Q. I hope you guys see how I did that. I just subtracted a Q, a Q from both sides. Anyway, that's how we ended up with that. So now we have 1 minus Q equals P. Again, we just figured Q out, so we plug that in. So 1 minus 0.01 is 0.99. So P equals 0.99. And then from there, now we have our P, right? Because we know that Q is equal to this. We know that P is equal to this. Therefore, we can figure out 2PQ, because all you do is multiply those two things together, multiply them by 2, and we get that. So we know that about 2% of the population are heterozygotes for this disease, meaning, and this isn't just trivial, this is good for public health, because that means we know about 2% of the population are carriers, right? We know who has the disease, obviously, because you have it, and it's one of those things you get diagnosed with. We know you have it, just like we would know if you had Ebola, or usually COVID, as long as it's not asymptomatic. This is one of those things, though, or this is also one of those where if you have it, we know it. However, just like most recessive things we've talked about, you have to have both alleles. Therefore, if you have only one allele, you don't have the disease, but you're a carrier. And that's an important thing to know. So when you're having children, you know your risks, so you have a better idea of what your risks are of having a child that has it. Anyway, we're done talking about that. To remind you, I'm not going to ask you to do any of this on the exam. I just wanted to show you how this big, scary looking equation can actually be used algebraically to figure things out with public health and why it's actually important. And it's not just like, I wonder how many wildflowers are red in that particular field. That's not that important, right? So any questions about that? Okay. Let's talk about microevolution as a change in a gene pool. Hopefully we can move through this quickly because you're already familiar with it from the lab. And here's some good news for those of you who did do the lab. As always, you can do it for half credit. Anything you missed, you can fix for half credit. And now you're going to get a good lesson on everything you should have learned in the lab. First of all, a non-evolving population is in what's called genetic equilibrium, which is also known as a Hardy-Weinberg equilibrium. Now, we didn't talk too much about this in the lab, but for those of you who did the lab, consider what you learned and what you understood about the lab, about how, remember, the population of those fish changed, right? The color, like was it mostly orange ones, mostly white ones, mostly orange with white spots? It all changed depending on what you did. But if they don't change, that is called Hardy-Weinberg equilibrium, AKA genetic equilibrium. And you need to know that. That's a word you need to know. I should have put that in bold. You need to know what genetic equilibrium is. You need to know that it also equals Hardy-Weinberg equilibrium. And again, this is when you have no change. It's when the gene pool remains constant from generation to generation. Another way to say that first bullet point, excuse me, let me get this going here. Another way to say this bullet point is sort of like this one. It's saying it in a slightly different way. The frequencies of alleles, your P's and Q's, right? Your dominant allele versus your recessive allele, and the genotypes, your homozygous dominants, your heterozygotes, your homozygous recessive, they are unchanging. So again, in Hardy-Weinberg equilibrium, nothing is changing. Those numbers do not change. Because generation to generation change in allele frequency is evolution on its smallest scale, this is what we call microevolution. So maybe that's another word I might ask you about, but probably not. I'm more involved or more interested in you understanding how microevolution works than the definition of it. But yeah, that's what we're talking about. Again, microevolution, we're not talking about this big change in species, right? Like where you go from, some wolf-like ancestor and that evolves into a dog, right? We're not talking about changing species. We're talking about the same species, just certain aspect of the species changing. So any questions so far? All right, let's talk about the mechanisms of evolution, and this is the part that you are already familiar with, partially, because you've been exposed to them in the lab. The next part. I'll give it a second, though, because I see some note-taking, which is a good time. That reminds me, today's word, the first word for attendance, is scale. Like you weigh yourself on a scale, or you remove scales from a fish. If you've caught it and plan to eat it. If you're just doing it for the fun of it, you're messed up. Anyway, the mechanisms of evolution. We're going to talk about, I was about to say other than natural selection, but we're going to talk about that when I get into it. But remember, again, evolution is a big, broad term. There's all these different ways evolution can happen. So far, we've only talked about natural selection. Now we're talking about the other ones. Again, natural selection is the most important, but the first one we're going to talk about is genetic drift. And you're already familiar with this. You've been exposed to it in the lab. You need to know what it is. It is the change in genes, or the gene pool. Whoops, there's a typo. Change in gene pool due to chance. So, a good opposite example of this would be sickle cell. That was a change in the gene pool, and it had nothing to do with chance, right? That gave you an advantage in places with mosquitoes. Therefore, that was natural selection. But this, what we're about to talk about, is just chance. And here we go. Let's visualize it. Here's a random flower, a field of flowers. Some of them are red, some of them are white, right? Those are the phenotypes. For the genotypes, we've got some of them are homozygous dominant, some of them are homozygous recessive, some of them are heterozygotes. Now, the thing about this color is there is no advantage to being red. There is no advantage to being white. There is no advantage to being homozygous dominant or heterozygous or homozygous recessive. No advantage at all. So by chance, these things of this population that happen to mate is the one that the author of the textbook drew squares around. So these are the flowers out of all of them that just happen to mate by chance. It's not because there was any advantage or sexual selection or anything. So anyway, those flowers mate, and these are the offspring. that whole thing. And then out of all those, 2 of those happen to mate, and they happen to be homozygous dominant. So by the 3rd generation, the entire field of wildflowers is all homozygous dominant, right? They're all red. And you can see the numbers right here, how the numbers change. Here we have about a 70-30 mix. Here we have a 50-50 mix. And here we've eliminated the other allele. And that's all by chance. Just an example. So I'm not going to ask you about red flowers versus white flowers on the exam. For the exam, what you need to know is that genetic drift is a change in the gene pool, and it's completely by chance. So there's no advantage to one genotype or phenotype over the other. Any questions about that? All right. That's a good time for me to put something on the board to help you keep track of what we're talking about. And I'm also going to abbreviate like I like to do. Again, we're going to talk about evolution. That's what this whole chapter is about, which is that's a great test question. I don't know how you guys keep missing that. So the first thing we're talking about for the next exam is evolution. That'll be a test question because I always thought those would be easy, but everyone keeps missing them. Anyway, under evolution, there's different types. The one we're talking about right now is genetic drift, right? That looks bad, GD. We're talking about GD right now, genetic drift, just a subcategory of evolution. Now, moving forward, genetic drift itself has 2 subcategories, right? So we can subcategorize things even more. And I hope you understand when I say by subcategorize. That's like saying if this was, today we're talking about vehicles. And the type of vehicles we're going to talk about are Fords. And then under Fords, we're going to talk about the F-150 and the Ford Focus. That's what I mean by subcategorize, right? Anyway, the first thing we're going to talk about under genetic drift is the bottleneck effect. I'm going to abbreviate, BN, bottleneck. This is an example of genetic drift, and this is a result from a drastic reduction in population size. And I'm glad your book led off on this, because if you think about this too hard, this could become confusing, and I hope you guys do think about things instead of just taking in the information and storing it away. I hope you're thinking about it. The thing that threw me off as a student is I'm talking about a reduction in population, but also we're saying that there's no advantage, right? So keep in mind that this reduction in population has nothing to do, like the black moths, for example. That would not be the bottleneck effect. Though you could say, there was a huge reduction in the population of the light moths, right? Once things got polluted and the birds started eating all the light moths. But that was natural selection because the light moths were at a disadvantage. This is when you have a huge reduction in population and it has nothing to do with genetics. And the reason it's called a bottleneck effect is because basically what we're saying is that the population is passing through a bottleneck. Have you guys ever heard that term when it comes to traffic? You've got like 4 or 5 lanes of traffic, and then it all narrows down to like 1 or 2. That's a bottleneck, right? So the same thing applies here. Now, the reason this is important is because it decreases genetic variability. So think about that example of the wildflowers. That wasn't a drastic reduction in population, but assume it was. Just remember, we did see a change in the population after 3 generations. We saw at the end that it was nothing but homozygous dominant, right? By chance. So this is kind of similar in that some alleles might be completely lost. Not because one allele was better than the other, because it just might have happened. Here's a decent example. It's a hypothetical because it actually uses a bottle. So you can see this varied population. It's got some blues, or maybe there's purples. Some purples, some greens, some golds, and there's marbles, right? That's the population, pretty decent sized population. Then you dump it out, that's the bottleneck, and only a certain few survive. And by chance, it was almost entirely purple with one green. And in this case, all the golds went out. So that's a bottleneck effect. And again, you can see that was all by chance, right? There was no advantage to being purple. There was no advantage to being green. There was no advantage to being gold. It just so happened that purple mostly won out on that bottleneck with a little bit of the green. Now, when we talk about later, I think next chapter, we're going to talk about biodiversity. And you're going to understand why biodiversity is so important. And when that happens, think back on this discussion. But for now, just trust me when I say biodiversity is important. You want as many different colored marbles as possible. Anyway, that's a hypothetical example because it's nice and easy to visualize. Here's a real example. Cheetahs. cheetahs have had at least 2 genetic bottleneck effects, and you might need. Normally, I say you don't need to know examples, but you might know this one. You might associate cheetahs with bottleneck. I might ask about that. Probably not. It's definitely on this study guide. But anyway, you don't need to know the details. I'll tell you the details. The first bless you. The first bottleneck effect that we know about was probably about 10,000 years ago during the last ice age. So you had this huge population, and then because of the ice age, many of them died off. So there wasn't not like the strongest survived. It's just it was so harsh that a lot of them just died, regardless of how strong they were. And then the 1800s happened again because farmers hunted them to near extinction. And again, I mean, this is a little bit iffy, right? Because you could argue, well, the cheetahs that were best at hiding from the farmers, the quickest or best at whatever it was. So you could almost argue that that's natural selection, but it was such a drastic reduction in population, and the farmers were being so, I don't know, persistent. Like they weren't being picky. They're like, we're going to kill as many as we can because they're killing our livestock. So yeah, two genetic drift bottlenecks. Anyway, with so much little genetic variability, they've reduced the capacity to adapt to environmental changes. So this goes back to what I was saying that we're going to talk about a lot in the upcoming chapters. Biodiversity is important. Think about all the humans that you know, either in person or from the news. Some people get COVID and don't even know it. Some people get COVID and have like a slow, miserable death. Some people get COVID and it's like the flu. That is genetic variability. That's because, well, there's other things involved too, but think of it that way though, right? Think we all have these different genes, right? And because of that, some of us are more susceptible to that one disease than others. And that's the same thing with the that's a good example of why having a high amount of biodiversity is good. So imagine if every human had the same genetics as those humans that die slow, painful deaths from COVID. We would be in trouble, right? Because if everybody was that susceptible to it as a population, then the human population would be decimated by COVID-19. But we have all this variability, therefore we can adapt to that environmental change. Anyway, any questions about the cheetahs? All right, the next thing we're going to talk about, we're still talking about genetic drift, which is a subcategory of evolution. The next one we're going to talk about is founder effect. And in a way, it's almost, depending on how you look at it, it's the same as a bottleneck. In that, you're going from a large population, cutting it down to a very small population, and the new organisms that make up the new population is completely by chance. It's not nature selecting them. It's not sexual selection. It's just by chance. These are the animals that made it. The Galapagos, think about that, even though we didn't talk about it much. Like I said, the Galapagos Islands are far enough away from mainland South America that there was very rarely any creatures going from South America to the Galapagos Islands because it's really far away. But sometimes a select few made it. So those select few represent a very small portion of that gigantic population on mainland South America. And those were the founders. So if there's like a population of birds that range from really dark black to really light brown, and then it just so happens that the really dark birds made it to the islands, then the new population is going to be mostly dark. Not because there's any advantage to it, just those are the ones that happen to make it. That was a hypothetical one. Your book gives some real examples, and they use human examples, and that's why I like it. If you download the PowerPoint, you can click on any of these links, and there's little short videos you can watch about the Pingalap people, the Amish, Martha's Vineyard. And what I love about these examples, well, I'll get back to that. Pingalap, those are people, I think it's definitely in the Pacific. I think it's the Southern Pacific, where the people who left mainland Asia and made their way, through the Polynesians, whatever, those people just so happen to have some colorblind people with them. Not a lot, but a higher population than the rest of the population of Asia, where they came from. So because of that, the people that live on that island have a higher population of colorblindness than the rest of the humans on Earth. And I like that, right? Because think about it, that's on an island in the middle of the South Pacific. Let me skip the Amish for now and go to Martha's Vineyard. Does anybody know where Martha's Vineyard is? It's It's like a little island off the coast of Massachusetts, as far as I know, and I don't know too much about it economically, but I think it's like one of those nice vacation destinations. It's a high class kind of place. But anyway, the people that founded Martha's Vineyard and this is back in the day when you just didn't have ferries and planes and you can go back and forth easily. The people who founded it's an island, I should mention that. People that founded Martha's Vineyard had a higher population of deaf people, a high percentage of deaf people than the rest of the people in the United States. I'm not saying it was a lot. I don't remember the exact numbers, but it was a higher percentage. And because of that, there's a higher percentage of people in Martha's Vineyard that are deaf. And that's a great example of how this is completely random and not natural selection, because there is no natural selection advantage to being deaf, generally speaking, right? So there was no advantage to being deaf. It just so happens that the people that came over to that island had a higher, there was a higher percentage of them. As a matter of fact, that's where American Sign Language was created, was Martha's Vineyard. Anyway, the two examples I just gave you are islands, and I like that because it's easy to visualize that, right? You have a large population somewhere else, and then a chunk of that population gets put onto an island. What I like about the Amish example is the Amish, they're not in a geographic island, right? They live, in Indiana, Ohio, places like that. But culturally, they are in an island. Where is it? Isolated habitat is how your book describes it, right? So in a sense, the Amish, at least reproductively, are in an isolated habitat because generally there's not a lot of people coming into the Amish community, right? There's not a lot of converts to the Amish way of life. So because of that, they're mostly inbreeding, which is also why you have, and again, if you click the link, you can see some examples, but they have a high proportion of a lot of diseases that you usually only see with inbred people because there's a lack of genetic variation and because of the idea of the founder effect, right? Whoever founded that Amish religion way back when, they had a higher percentage of these certain diseases that you can learn about if you click the link. Any questions about the founder effect? Pretty simple stuff, right? The next thing we're going to talk about is gene flow. And I love this because this is in the name. It should be easy. And again, you're going to need to know, if I give you an example, most likely that's what will happen in the exam is I'll give you an example of something and you'll be like, oh yeah, he's describing founder effect or bottleneck or genetic drift in general, or as we're about to learn, gene flow. And when I say you need to recognize an example, what I mean is I'm going to come up with some other random example. It might not even be a real example. So you don't need to go studying all the examples of genetic drift or gene flow. Just be able to recognize a hypothetical one if I give it to you. But gene flow, anyway, this one is in the name. And in a way, you almost could describe it as the opposite of founder effect. Gene flow is the genetic exchange with another population. So remember, populations, it's a certain species, right, that are all interbreeding. And for the most part, they're not breeding with anybody else. Remember, if you get in your mind, picture that last lecture we had, and there was those lakes, and I was talking about fish and how those fish aren't going to go from one lake to the other, mostly, but sometimes it might happen. Same here. So again, think of those lakes. I'll try to draw it. So you have all these isolated lakes. And mostly, fish don't just get up out of the water and take a walk to the next lake. So these are isolated populations. And these, right, there's an isolated population. Here's another one. Here's another one, right, in their own lakes. But occasionally, you might have, an eagle swoop in and grab a fish from here, fly off, and accidentally drop it into there. That's gene flow. Because those were two previously separated populations that almost never exchange genes, but sometimes it happens. And that is gene flow. And you need to know that for the exam. Gene flow is the genetic exchange with another population. And it's in the name. It's when the genes flow. To me, the hardest part of this, when I took this as a freshman, was differentiating, at least on the exam, between gene flow and genetic drift. Because in my mind, I kept thinking, all right, genetic drift, that's when the genes drift from one population to the other, right? So try to be careful with that, because that got me for the longest time. Genetic drift in a vacuum is an easy concept to understand. So is gene flow. But then when you put the two together on an exam and you need to differentiate between the two, it can become a little bit challenging. Anyway, so gene flow, again, it's exchanging of genetic material, of genes. Your book points out that it usually does reduce the genetic differences between populations. So again, and I use this example when we're talking about natural selection. We had all these different ponds, and I said the fish in this pond have different circumstances than the fish in this pond, right? This pond might be really shallow. This one might be really deep. This one might have a lot of green algae. This one might have a lot of brown algae, right? All these things that might cause, even though it's the same species, this one might become, I don't know, bigger and browner. These might become smaller and greener, like the fish, right? And that would be natural selection. However, and again, those allele differences would be different. But with this, you can add some more brown alleles to the green population and some more green alleles to the brown population, or small alleles to the big population, or big alleles to the small population. Point here is that, generally speaking, genetic or gene flow can be a good thing because it increases biodiversity. Any questions about gene flow? All right, now here's the easy one because we've already talked about it and it's the big one. Natural selection. So we're looking at it again. And also that's it. When it comes to evolution, we talk about genetic drift, gene flow, and natural selection. So let's jump back into natural selection. A closer look. So of all these things that we've talked about, it's only natural selection that leads to an adaptive evolution. What is an adaptive evolution? It is evolution that results in a better fit between an organism and their environment. So again, in this goofy little example, like I said, if there's a lot of, I don't know, green algae in this lake, then maybe over time, those fish will turn more green because that provides them with more camouflage, right? Or the dark moths, from the lab you did. Your book is another example. Obviously, it's just an example. You do not need to memorize the details of this example because just like everything else, for the exam, I'll probably give you an example you've never had, and you need to recognize that, like, yeah, that's natural selection. But anyway, the blue-footed booby, it's an excellent example of an adaptation for its home. Why is that? Well, let's talk about it. And I can show you a picture of it here in a second. Its body and its bill are streamlined like a torpedo, minimizing friction as it's dived. So what this thing does, the way it eats, it flies around, it's looking for its prey, which are fish, and when it sees it, it just dive bombs right into the water, and that's how it goes fishing. So obviously, the faster it can glide through the air and the water, the better it is. It has a large tail that functions as a brake. Well, that's a good thing because evolutionarily, we know, like I just said, that it's streamlined to go slice through the water. So obviously, because you're a bird and not a fish, you don't want to get too deep into the water because you can't breathe underwater. So it's a good thing that once you catch your fish, you can put on the brakes, right? Slow yourself down, get back up to the top of the water. And I don't know why your book chose this example, because this should be discussed at another time. But females prefer males with the brightest blue feet. And that is not an example of natural selection. That is an example of sexual selection. Whoops. And I lied to you right there. We'll talk about that later too. There is one more category. I'm sorry, guys. Sexual selection. And we'll talk about that later. Anyway, obviously you can see the natural selection portion of this, right? The ability to get its food and stop from drowning. The better you have that ability, the more you can eat, the more you can survive, the more you're going to reproduce, the more you're going to pass those genes along, and therefore the population over time is going to have a more streamlined body and bigger tails that function as brakes. Any questions about that? The second word for attendance today is booby, since we're talking about it. Like a blue-footed booby, it's booby. Here's a picture of a blue-footed booby doing its little dance. You can see it's streamlined, right? You see that tail, that really large tail that functions as a brake. Those blue feet. And again, we'll talk about that here in a second because of sexual selection. Now, I don't know if I want to do this. Let me think about this. I don't want to confuse you. For the exam, I'm not necessarily going to need you to understand whether sexual selection is a part of natural selection or a subcategory. Don't worry about that. The only thing you need to worry about as far as subcategories is concerned is genetic drift. Right? Genetic drift has two subcategories, bottleneck and founder effect. Everything else, you just think in your mind, all right, this is all different examples of evolution. And the reason I say that is because sexual selection, you could argue, is a type of natural selection because Actually, does anybody know why a female might prefer a blue foot? Because obviously there's no, as far as normal thinking is concerned, there would be no advantage to having a blue feet for survival.
Strong genes.
Genes, good. That's what causes, the blue foot. Yeah.
Increase of offspring.
Yes. Yeah, this is great because you guys understand the big concept. So specifically, I'll tell you, and this is a hypothesis, I think, or maybe they do know. Usually when you see birds, especially with birds, when they prefer like some kind of like really fancy plumage or really bright colors, there's no necessarily survival advantage to that. But for them to have a really bright footed booby, not only do they have to have the right genes, but they need to be healthy. So if there's some blue footed booby male out there that's got parasites and it's just not really good at getting food, so it's starving to death, it's probably not going to have really bright feet. It's probably going to have really dull colored feet. And the female's going to be like, nope, I don't want that. So you could argue that that's a form of natural selection. But anyway, we'll come back to that. I get too excited about evolution. I jumped the gun. Anyway, let's talk a little bit more about evolutionary fitness. And here's a word you should know: relative fitness. And I think when it comes, you might just think of this as fitness. I mean, yeah, technically this is relative fitness, but I don't think we're going to talk about many different types of fitness. Just fitness in general. It's the contribution an individual makes to a gene pool relative to the contribution of other individuals. So in that first evolution lab you did with the moths, the black moths had a larger contribution to the gene pool than the light moths, because the light moths mostly were getting eaten. Therefore, they were not reproducing. Therefore, they were not contributing to the gene pool. The black moths mostly were not being eaten. So they were reproducing. So they were contributing to the gene pool. Your book points out that sometimes this is called slightly inaccurately, the survival of the fittest. But not necessarily the fittest. In these examples we're giving, yes, it is. But if you were to really look at it, there's other things that don't necessarily fall into that category. But anyway, when you think of fitness, again, we're just talking about who's putting more genes into the gene pool, right? Who's contributing more to the gene pool? That's all that means. So any questions about that? Relative fitness? All right, the next thing we're going to talk about is the 3 general outcomes of natural selection. And I hope this is easy. To me, this is one of those few concepts when I first learned about evolution or natural selection that was easy. Is that basically there's only 3 things that can happen with natural selection. And that's what we're going to talk about. And we're going to use mice as an example. We're going to graph a population of mice. And we've already said this before when we were talking about chapter 8, was it? When we were talking about polygenetic inheritance, where there's a lot of genes, polygenic inheritance, where there's a lot of genes contributing to one phenotype. Same here. If you graph the population, there'll be a bell curve. Let's just look at it and I'll show you what I mean. Actually, let me back up. Yeah, see, this is the writing. I'm not even going to talk about it because I'm just going to show you in the picture. So here's the original population, right? There's really light colored mice, really dark colored mice, and everywhere in between. And what this graph means, I hope you guys understand what that means, right? You can see up there at the top left, frequency of individuals. That means that there's very few white ones, very few dark ones, and there's a lot of those middle ones. You guys see that, right? And then from there, there's, like I said, there's most of these, there's fewer of these, and there's even fewer of these. Most of this, fewer of these, and even fewer of those. That's what a bell curve means. Now, there's 3 different ways that evolution or natural selection can change its population. The first one that you need to know is directional selection, and it's in the name. Specifically, when we say directional, what we mean is we're moving the direction of the bell. So the bell can either go left or right. Meaning, all right, let's say this population of mice lived, on a mountain with a lot of different colored rocks. Light colored rocks, dark colored rocks, and medium colored rocks, and everything in between, just like the mice. So because of that, let's just say most of those rocks were this light, middle colored gray. So because of that, those mice have the best chance of hiding themselves and reproducing. Then let's say there's a rock slide or a volcano or something, I don't know. Something happens, and all those different colored rocks, they change. So now, instead of having mostly gray rocks, you have mostly dark colored rocks. So yeah, let's say there was a volcano. So now you have mostly dark colored rocks. Therefore, now those darker moths, those darker mice have an advantage, right? So now it's going to shift. There's going to be a direction that bell curve shifts. So there's going to be mostly dark ones and even fewer light ones. Does that make sense? Or let's say the opposite happened and there was a new ice age and everything was covered in snow. Then it's going to still shift. It'll just shift the other way. So then you're going to have a lot more light ones because they can camouflage themselves in the snow. That is directional selection. Going one way or the other, right? One extreme phenotype or the other. And I will give you an example of that for the exam. One that you've never heard of. I probably just make some stuff up like I did on the last exam. Like a population of aliens. So this population of aliens went from this to this. Is that an example of directional selection, disruptive selection, or stabilizing selection? And you'll need to recognize it. And again, if it goes towards one of the extreme phenotypes or the other, that is directional selection. Any questions about that? All right, the next example is this one in the middle, B, disruptive selection. So instead of going from one phenotype extreme or the other, this is going from one phenotype extreme and the other. So in this hypothetical example, let's just put together what I said for both of these. Let's assume that there was a volcano, so all the rocks are black now, but there was also an ice age, so there's a lot of snow. So what do you have? You have a lot of dark rocks, but you also have the white snow. But you have very few gray colored rocks. So because of that, being gray is really at a disadvantage now. But being really light colored is an advantage, and being really dark colored is an advantage. So because of that, the population shifts, right? You lose that medium phenotype, and you start going towards both of the extreme phenotypes. Does that make sense, everybody? And again, for the exam, all I'm going to do is give you an example of a population that went where it lost its medium phenotype, right? It went towards the extremes. A real life example of this, and I wish we were still doing independent work because it's real fun to look it up. There's a beetle out there, and I think it's called the sneaky bastard effect, where these beetles fight to mate, right? So the bigger the male is, the more likely it is to mate. However, when the two alpha males start going at it and fighting, there's also the small males that look like females. So the two are fighting, and this little small male just walks right past them. They don't even notice because they're fighting. And he goes and has sex with the female, and he's out. So in that case, there's an advantage to being bigger because the bigger one's going to win the battle probably and probably mate. But also, the smaller they are, the more they are to look like females, in which case they can sneak past and, again, pass along their genes. Anyway, any questions, again, about directional selection or disruptive selection? The last one is very simple. It's the logical conclusion to what we're talking about. It is stabilizing selection. So in this case, you don't go towards one phenotype extreme or the other phenotype extreme. You don't go towards both phenotype extremes. Far from it. It's the opposite of everything we've just said. This is when you get even fewer of the extremes and even more of the middle. So again, forget about these two. We're done talking about those. We're just looking at these two right here. So the original population had very few light ones, very few dark ones, mostly gray ones, right? It's mostly this. And you can see, because of whatever happened, now there's even more of those gray ones, and even fewer dark ones, and even fewer really light ones. That's just stabilizing selection. And a human example of that is the human head, or human birth weight. Obviously, if a baby's too small, it's not going to have enough resources. It's more likely to die. Of course, we have modern medicine to help with premature babies and smaller babies. But generally speaking, especially if you got rid of natural medicine, if you had a really small baby, it has the less chance of making it. And if you have a really big baby, the mother and the baby have a little less chance of making it because you have to birth the thing. So because of that, humans have a pretty strict range of how big and small they are. Again, before modern medicine, because with C-sections and things like that, it's a game changer. Actually, there's debate too on how C-sections and modern medicine have changed that stabilizing selection. It might be unstabilizing. Anyway, any questions about this? The 3 different outcomes of natural selection? All right, let's move to the last thing we're going to talk about in this chapter. Thank goodness, because we are behind. only by a day. But we've already talked about it on accident because I got excited about it and started giving you a preview. But the last thing we're going to talk about is sexual selection. And the definition is that is when individuals that have certain traits are more likely than others to mate. And that brings us to another word you need to know, sexual dimorphism. That is when there's a distinction in appearances between males and females. So birds are a great example. You see those really dull colored female cardinals versus really bright male cardinals. Some plants are good examples. Some are not. Of course, some plants are both male and female at the same time. But anyway, it's pretty simple stuff, right? Sexual selection is when there's something about you. Pardon my French, but this will be easy to remember this way. There's something about you that gets you laid more than everything else, more than your competitors. So any questions about that? It's pretty simple. I'll give it a second. The next slide we're going to talk about is we give some examples. But yeah, it's pretty easy. Obviously for the exam, for your hypothetical example, I'm going to say, this, I'll probably do female because it's usually a female choosing. Actually, while I'm waiting, any guess why it's usually the female choosing the male? And it is almost like that. Or it is like that almost with every example of sexual selection is almost the female choosing the male. Any idea why? Yes.
I say because she wants the babies to have a good chance of surviving.
That is true. You could also say the same for males, though. Could. Basically, it's because of this. Like, think about how many sperm men, well, let's just use humans for example. Think about how many sperms men produce and put out there versus how many eggs women produce and put out there, right? Women have one a month that they put out there. Men, it's almost unlimited how many sperm we can put out there in a month. I would love to know the numbers. Actually, I wouldn't love to. I'm curious to know the numbers of how much sperm a man can put out in 1 month. And that's just the one thing. That's just the cell. Then you have to take into consideration, usually it's the female, not just with humans either. It's the female that is taking care of that new developing life, right? Some species more than others, but the male can be like, hey, here's my sperm. Good luck. And with humans, that's obviously a bad thing. But a lot of organisms, that is exactly what happens. Here's some sperm. Have at it. Do what you will. So anyway, that was just to waste some time. Let's talk about this next slide. Some species have secondary sex structures, which are used to compete with members of the same sex. Again, it's usually males. Some of these secondary sex structures are used in combat. So like horns. And usually, or sometimes is often used in ritualized display. Does anybody know what this picture is on the right?
Peacock.
Yeah, it's a peacock. That's the male peacock. Obviously, because the female is called a peahen. But that big old tail, that's the thing. Again, pardon my French, but to help you remember it, maybe that's the thing that gets it laid. The female looks at that tail, like, shake your tail feather. That's what I'm looking for. But here's an interesting, the reason I like this example is think about this. That is disadvantageous to survival. So for natural selection, you don't want a big tail. In India, that's going to get you eaten. That's going to get you noticed by a tiger. It's going to be harder to fly away from a tiger. That's a bad thing for natural selection. But for sexual selection, the females are all about it. And again, like we already said, it's usually the females that are choosier than the males. We already talked why. Females have more invested in producing new offspring. Male are just like, here's one of my mini sperm. Do what you will with it. There's some picture examples of what we're talking about, some sexual dimorphism. On the left, the female bird is on the left, the male bird is on the right. And then, of course, we can see some bighorn sheep or whatever competing for males. Now, that brings us to the end of the chapter, which is evolution connection, which we have in every chapter, and obviously we would have it in the evolution chapter. Has anybody ever had to take antibiotics and the doctor's like, make sure you finish all 10 days or whatever, right? The reason being, and the reason this is so important, and this goes back to that pesticide discussion we had at the beginning of the chapter. When you first start taking antibiotics, you'll feel better like almost right away because those things work and you're killing off all those weak bacteria. But what's happening is you're killing off the weak ones first. And because most of the infection is gone, you feel better. But there's still some strong ones in there that haven't died yet. So if you quit taking it, what you've done is you've killed off all the weak ones and only the strong ones are left. And if they get a chance to reproduce, then you're going to have a worse infection. Anyway, the last word for today is going to be soccer. As you can see the picture of it right there, soccer. And that's it. I'll see you guys on Wednesday. I'll be online for office hours. Hope you have a good week.
The number is, based on that, the Great Sperm Race.
Oh, that's right. Yeah.
If it's once a day, and it's 15